Dear scholars and the Science Father organization, hello. It's my great honor to have this opportunity to share my research with you. The topic of this study is response of soil aggregate-associated microbial and nematode communities to tea plantation age. As the primary regulators of decomposition, soil bacteria and fungi form key linkages between detritus and soil fauna, and comprise the principal resource base for soil food web, maintaining pH. Decomposition and diversity of the soil microbial community is critical to sustain soil ecosystem function and to mediate soil organic matter decomposition and nutrient cycling. Has a central role in the soil food web. Soil nematodes are believed to be tightly associated with microbes both functionally and nutritionally. In consideration of the trophic interplays, nematodes are assigned as bacterivores, plant parasites, fungivores, and omnivores. Anti-predators. Assessment of nematode community composition can provide unique insights into soil biological processes because different feeding groups of nematodes are specialized with respect to their food sources and play essential roles in functioning of soil ecosystems. Thus, measurement of soil microbial and nematode communities together is helpful to not only complement our understanding of the dynamics of soil food web, but also indicate the effective need, assess management intervention as well as the developmental status of restored ecosystems. To understand their variations in agricultural soils, the communities of soil microbes and nematodes should be recognized at different spatial scales. As the basic unit of the soil structure, aggregates serve as the heterogeneous assemblages of the mineral and organic particles. In the meanwhile, they are classified according to size such as micro 0.25 millimeters and macro 0.25 millimeters aggregates. The microaggregates are produced through the microbia, mediated processes in the macroaggregates, which depend intensively on the structural stability's persistent organic binding agents. Conversely, the temporary associations of particulate organic matter, minerals, and microaggregates finally lead to the production of macroaggregates, which are perceived mainly through the enmeshment by fungal hyphae and plant roots. Besides access to food sources, the fundamental requirement for soil organisms is adequate space to accommodate their growth and movement and to allow for water and gas exchange. For example, soil bacteria can occupy a pore less than 3 m in diameter, whereas larger organisms, such as soil nematodes, are limited to larger pores. As a result, the concentration and availability of organic substances, as well as the chemical and physical protection mechanisms, dominate the distribution of soil biological communities within different aggregate fractions. Poor land use practices resulted from increasing human population pressure over the last century, including deforestation, overpasturing, and overcultivation, have drastically increased the fragmentation of ecological environments in the hilly area of western Sichuan in China. This has led to not only the intense degradation of natural ecosystems, but also the reduction in the current and future soil productivity capacity as a result of windban, deep water erosion, declines in fertility, alterations in moisture and aeration levels, or shifts in soil fauna and flora. To ameliorate the situation, a series of nationwide conservation projects dedicated to rehabilitate these ecosystems have been implemented by the Chinese government. Among them, one focus for the sustainable agricultural development of the hilly area of western Sichuan is to convert the abandoned land to tea, camellia sinensis plantations. As an important cash crop in many developing countries, tea is widely cultivated in Sri Lanka, India, and China, particularly as the largest tea producer all over the world. China had the area of 2.93 million hectares used for tea plantations in 2022, which has been continuously increasing. Our previous studies have found that long-term tea cultivation significantly influences the distribution and stability of soil aggregates. However, the changes of soil biological communities still remain unclear. Therefore, the proposed study is designed to determine the short and long-term influence of converting abandoned land to tea plantations on the dynamics of soil microbial and nematode communities associated with aggregate fractions. It is hypothesized that the microbial and nematode communities within soil aggregates would differ with the size of aggregates due to resource and space constraints within each fraction. Two soil microbial and nematode communities would differ with the age of tea plantations because there would be significant changes in the distribution and stability of soil aggregates during the tea planting process in the hilly area of western Sichuan and Sea. Heal. The results and discussion include the following 3 parts: soil aggregate associated total PLFA, an indicator of total microbial biomass, significantly declined as the particle size decreased, with a significant increase in small macro aggregates compared with medium macro aggregates in all the tea plantations. Similar trends were observed for almost all the microbial taxonomic groups, including bacteria, fungi, and actinomycetes. In the macro aggregates, total PLFA and PLFA associated with different taxonomic groups were significantly higher in the 23-year-old tea plantation relative to other plantations. In the micro aggregates, however, there were no significant changes over time. Soil nematodes in this study were dominated by bacterivores, followed by the plant parasites, cumulatively representing approximately 90% of all the nematodes identified. Aggregate-associated total nematode abundance significantly decreased with decreasing particle size regardless of tea plantation age. Additionally, the abundance of different trophic groups, including bacterivores, plant parasites, fungivores, and omnivores and predators, appear to show a trend similar to the abundance of total nematodes within different aggregates. In the macroaggregates, the abundance of total nematodes as well as different trophic groups, with the exception of fungivores, was significantly higher in the 23-year-old tea plantation compared to other plantations. In the microaggregates, on the contrary, no significant differences were observed among the different age tea plantations. Soil microbial biomass, including total microbes, bacteria, fungi, and actinomycetes, was not correlated with most nematode abundance across aggregate fractions in different tea plantation ages. In the macroaggregates, the biomass of soil total microbes as well as different taxonomic groups was positively correlated with most nematode abundance, especially total nematodes and bacterivores across tea plantations of varying age from 16 to 53 are old. In the microaggregates, however, no significant correlations, p 0.05, were observed between microbial and nematode communities. As discussed above, not only microbial biomass and nematode abundance but also their diversity was highest in the large macro aggregates regardless of tea plantation age. However, a shift occurred from large to small macro aggregates after 23 years of tea cultivation, leading to a decrease in large macro aggregates in the whole soil, which could also partly explain the degradation of soil microbial and nematode communities after 23 years, as indicated by the decrease of microbial biomass, nematode abundance, and their diversity. As presented in a previous study, the large macroaggregates represented about half of the whole soil mass in all the tea plantations. Meanwhile, we also observed that the 23-year-old tea plantation was enriched by the large macroaggregate and had more stable soil structure than other plantations. According to the aggregate hierarchy concept, the amount and quality of crop residues returned into soils might determine their distribution in different size fractions. In the early stage, the litters had a relatively high availability to soil microorganisms and consequently could be easily incorporated into large macroaggregates during their decomposition, contributing to the formation of large macroaggregates. In the later stage, on the contrary, the gradual processing and humidification of litters was responsible for the transition from large to small macroaggregates. Additionally, the reduction in litter amount and coverage after 23 years of tea cultivation enhanced rainfall alleviation and human disturbances by pruning tea plants and fertilization, thus causing large macroaggregates to break down. Thus, despite the distribution patterns of aggregate-associated microbial and nematode communities were mainly governed by the size of aggregate fractions, the age of tea plantations played a critical role in influencing the formation and stability of soil aggregates, implying that the different ages of tea plantations did not influence the soil biological community dynamics in isolation, but rather, the changes of soil aggregates were fundamentally important in this process. Conclusions. Bacterial pathway was a key to organic matter decomposition in soil, and bacteria dominated in microaggregates, 0.25 millimeters. Furthermore, higher stability of soil food web was observed in large macroaggregates, 2 millimeters. Strength of bacterial pathway was increasing after 23 years of tea planting. Tea plantation of 23 years improved significantly stability of soil food web compared to other tea plantations. Soil lobularity and pH exert stronger effects on the abundance and diversity of microbes and nematodes. And the pathway of organic matter decomposition played a role in altering the stability of soil food web. End of report. Thank you for your attention.